Okay, so it's just gone on the hour. Um, yes, good morning, good evening, good afternoon, everybody, and a very warm welcome to you all. Pardon for the noise. Wherever you're joining from around the globe, and a special warm welcome to our guest speaker of today, Dr. Marco Zonta, who's joining us from warm and sunny Brazil. So, our webinar today is actually our first webinar of the series this year, and we'll be discussing the future of cervical cancer screening. And Dr. Zonta will give us an in-market experience and share some very nice cases from our MyLab cervical device, our Bethesda-based diagnostics device for cervical cancer screening. So, my name's Monique Dalton-Kishlat, and it's an honor and a great privilege today for me to moderate this webinar on behalf of NOUL. And I'm the global product lead for MyLab Cervical at NOUL. And just before we kick off, just a logistical topic. At the bottom of your screen in the middle, you should see a Q&A icon. If you can't see that, then you need to go into the three dots into More, and you will find it there, and you can drag it into the toolbar. That will allow you to open up the box and then type in any questions that you have during the webinar. We will try to answer as many questions as we can, but of course, if we're inundated by questions, we will follow those up after the webinar. But we will have a Q&A session at the end where we'll open up the dialogue for you to ask your questions to Dr. Zonta. If there's any other questions on a more technical basis for the NOUL team, we will be happy to answer those for you as well. Next slide, please. Yes. So just to give you an overview of what we'll cover today, I will give an introduction onto NOUL, about NOUL, who we are, what our mission is, why we're doing what we're doing. And then I will introduce Dr. Marco Zonta and hand over the webinar to him. He will then introduce his project in Brazil using our MyLab Sir device, the results, and he's got some very nice cases to present to you as well, and also introduce the value of this device in the Brazilian market, what the clinical application is, and how he's been helping the communities of women in cervical cancer diagnostics in Brazil. And last but not least, we'll open the floor to your questions and answer your questions that you have for us. So, about NOUL, who are we and what do we do and why are we doing what we're doing? So, at NOUL, our mission is to explore the global challenges that threaten human life and, of course, discover potential solutions for these problems and realize these possibilities. So, this is the real driving force behind why we're doing what we're doing at NOUL. And already we have three key products in the market, one for malaria diagnostics, one for blood analysis, and the main star of the show today, of course, along with Dr. Zonta and his experience, our cervical cancer screening device. So we launched the company the company was launched in almost or just over 10 years ago now in 2015. And almost 4 years ago, we were listed on the South Korean stock market, the KOSDAQ. And our CEO and co-founder, Chan Yang or David Lim, along with his co-founders, have been very integral in bringing this mission to life. And in addition, our NOUL family, our NOUL team, which is now just over 150 strong, smart individuals, are really bringing this mission into life where for cervical cancer, we're really supporting the community and helping tackle cervical cancer in the globe. So you can see a building here of our headquarters and also our research and development manufacturing plant. These are located just southeast of Seoul. Yes, most of our team are actually based in Korea. In Yongin-si is the name of the town. And yes, and then the rest are actually located in Europe and Latin America. So just to give you an overview of the last 10 years and our road to success. So we've achieved our ISO certification, so 13485, as well as our GMP, our Good Manufacturing Practice, along with CE mark, and most recently our MD-SAP. And we're continuing on with other global certifications and also local registrations. So you can see a list here of the countries where we're actually in process of registration. And if there's a country there or you're from a country that's not listed there, feel free to reach out to us in order to kick off the registration. In terms of global recognition, you can see some big names there. So we've won awards with the Gate Foundation and awarded the grant also for the Wright Foundation. We're working with NVIDIA. We have an on-device partnership. And for MyLab Cervical, most recently in 2024, we were listed in the UNITAID report as an upcoming technology to tackle cervical cancer. And last but not least, of course, we can't do this alone. We have strategic partnerships around the world with IVD companies, with pharma innovators, and also key customers such as Charité and Limbach Group in Europe. And so you can see the last 10 years have been quite busy and successful, and we hope to continue the trajectory moving forward. So just a little bit more detail about our three products. As I mentioned, we have a solution for malaria diagnosis, and we're using always a gold standard staining. So for malaria, we have Giemsa stain, and we're able to do digital microscopy and species identification with our MyLab MAL device. For MyLab BCM, we're able to offer a 5-part differential along with RBC and also platelet parameters. And again, we're using the gold standard Wright Giemsa stain. And last but not least, our cervical cancer device, MyLab Cer, where we're using an automated PAP staining. We're doing automated staining, digital imaging, AI analysis, and also reporting out, all full walk away, and all using Bethesda-based reporting system. So in summary, over the last 10 years, we have partners in 100 organizations across 25 countries and research and operations in over 20 countries. As I mentioned, our HQ is in South Korea, where most of our team are. We also have a regional office in Switzerland, and we have teams, product management and business development teams on the ground in Europe, in Germany, the UK, and France, and also in Latin America, in Mexico and Brazil. So with no further ado, I would really like to, and it gives us great pleasure to introduce to you Dr. Marco Zonta. Dr. Zonta has over 35 years experience in the industry in cytopathology with a focus on women's health and research. So a very warm welcome to you, Dr. Zonta. As you can see, he has a PhD in infectious and parasitic diseases from the Federal University of Sao Paulo. Specialization, of course, like I mentioned, in cytopathology with expertise in cervical cytology. He's the associate researcher for the Department of Infectology. He's the research group leader in molecular oncology, HPV, and STI, or sexually transmitted infections, as well as the CEO, founder, and CSO of Sageza Ecosystem. So quite impressive credentials there, Dr. Zonta. And again, thank you so much for joining us today and sharing your experience in market with our device and some cases that you've got. So I'd love to hand it over to you now. Thank you so much.
Thank you so much, Monique, for my introduction. And hello, everybody. Good afternoon from Brazil. It's a sunny day here in São Paulo. The temperature is around 32 degrees. Look like our webinar is so hot. First, I wanted to please to be here today to discuss about AI in cytopathology diagnosis. I want to like to say thank you to Monique and to the NOUL group to invite me to participate this webinar. I will focus my presentation in 3 steps. First one, presentation of flow of my lab's work. Second, presentation the results about 6 cases of cervical cytology. that we can identify the importance to use an EA in confirm results. And third, the application of the methodology in point of care in medical practice routine in the public health programs. Next one, please. Well, one of the main points to discuss today is they use the AI in diagnosis and as an important tool to a medical practice, not for replacement the professional, but is a tool to help in a medical practice. We have 4 important points to discuss in the first time in this presentation. The imaging slides analysis. There is an important system to coordinate. Oh, sorry, can't see the slide. Oh, sorry. Is the important system to coordinate the preparation, identification, the images for analysis with a greater definition and the images for the microscope. The second point is the early cancer detection. EA always for the better definition of the morphology characteristic cells, enabling the identification of pathological process and their early stage. The third point, and in my opinion, the most important, is reduce the falsus negative. Is a tool to definitions of the cell structures and morphology. It provides a great efficiency in cytopathological diagnostics for the professionals. And the last one is clinical decision support. It's an excellent tool to aid in decision making process for cytopathology diagnostics. Next one, please. The important thing is breaking barriers. And I think we have 4 points to understand when you use EA diagnosis. First one is integration and the costs. Cost is very important when you introduce a new technology in your routine. The algorithmic basis. This is a risk. This is a risk as EL system will capture basis in training with basic data tests. The third point is the ethical and regulation issue. The ethics in Brazil or the ethical committee here in Brazil in our world is an important system to classify the importance when you use new technologies. And the excessive dependence of use a new technology, not to help, but to define the case. This is a risk of the back, please. This is a risk that health professionals will become overly depending of this technology. Next one, please. Well, to validate this technology here in Brazil, our premise were sustained by 2 points. First, analysis of MyLab as a high precision diagnostic tool. And second, identify the lesion marker and the characteristics of the IA features. We use 10 samples to validate this equipment here in Brazil. First step, we prepared the 10 samples using the manual path and using the coplast equipment. It's equipment that built smear in the slide. And then we use the manual PEP stand to stand the cells and submit to 2 cytopathologists to microscope analysis. The second step, we use the MyLab set and use the automated scanning and the LA analysis to identify the possible changes in the smear. Then we compare both diagnoses using the microscope techniques and using the EI methodology. Next one. Well, the principle of this method to use MyLab First, you need to prepare the stain using a LBC cytology and use the cells preserved in liquid-based medium. Then you input the cells in the cartridge of my lab, who will process the staining and analysis the imaging. And the last step, you need to analyze this margin as my lab offered to you in the analysis. Next one. The most important thing in this methodology is innovation. The MyLab used the solid base staining technology that overcame to the limits of traditional base liquid stain. One important thing, my lab doesn't need a big space to be used. The technique has a fast and clean staining without require larger space or dedicate infrastructure to prepare the samples. Next one. The AI, when you use my lab, the techniques will be identified some fields as possible cervical changes and present to the analyst with imaging options but based on the Bethesda classification. The methodology was qualify the possible degrees of cellular alterations by the group, as you can see in the left side in the slide. Next one. Now I will present the results about our project here in Brazil. Next one. This is the methodology and background that we use here in the project. Microscope and AI-generated analysis were performed by two senior cytopathologists. I was one of them to analyze the both types of the imaging, the microscope and the MyLab imaging. The results were classified according to the Bethesda System 2024. The cytomorphological description and comments were presented on case-by-case basis, detailing and the images in EA performed by lab server. And the cases are described individually. Next one. Our first case is case 253. As you can see, we have some mature cells that we have a little or a mild dyskaryosis and a mild hyperchromy in the nucleus. If you can see, the morphology of the cells and the nuclei are preserved, but the atypical nucleus is showing the characteristic of this cytopathology case. The classification of this cytopathology case is LCU, low interepithelial lesion. Next one. And look the importance to use EA in the identification of possible cellular change. The analysis did by microscope classify the smear with a negative for intraepithelial lesion and neoplasia. But when you use the EA, we can identify a low-grade squamous intraepithelial lesion. My lab was performed and showed the mature squamous cells with atypical nucleus. The focus of the analysis of my lab were mild dyskaryosis, mild hyperchromasia, well-distributed chromatin, sometimes present in small clumps, regular envelope, but important to identify the choriocyte cells in the smears. Is very important to be the difference between LCU and negative for the end of the diagnosis. Next one, please. In this case, you can see a typical inflammatory reaction. As you can see, mature cells from the intermediate layer of the epithelium. And more important, my lab can identify repair, process, and endocervical cells or glandular cells in the smears. Next one. Both diagnoses were NOUL or negative for intraepithelial lesions and malignancy, but the important appointment is that my lab can identify just increase the nucleo compared to the scariosis. It is a characteristic very important to do the difference between negative, atypical, or positive cells. In this case, my lab can identify a regular perinuclear halo, different the coilocytes. It's important to do the diagnosis negative from the atypical nucleus. And more important than my lab can identify very well characteristic as a glandular cells. It's important to show the difference between squamous epithelium and glandular epithelium. Next one. In this case 2P21, you can see more mature cells from the intermediate layer from the squamous epithelium, but as you can see a number of nucleus with mild and moderate dyskaryosis. My lab can differentiate the size of these nucleus, and this is very important to classify what grade or what type lesion is in the smear. If you can see here, you can see a nucleus with high mild and moderate size, and the chromatin and deposit of chromatin near the nuclear membrane. Next one. This is the characteristic that my lab can identify when they run the process. Mild and to moderate dyskaryosis, mild hyperchromasia, well-distributed chromatin, but sometimes persisting small clumps or chromatin deposits in the nuclear membrane. The nuclear envelope is okay, and in a regular perinuclear halo, classify as not a coilocytes. In the same, one important thing, my lab can identify LCU when we did the microscope analysis. We classified this sample with negative or intraepithelial lesion. Next one. This is a typical case of LCU C3P69, and my lab could identify an important nuclear morphology. It's a multinucleation, if you can see in the slide 2 and the slide 3. It's important to show to the cytopathologists what the grade or what the size of the lesion. This slide was classified with LCU2. Next, please. And the microscope classification is equal to the my lab classification. One important thing is my lab can identify another coilocytes in this smear. Next one. Case 3P13 is a lot of coilocytes identify from my lab. It's important to identify these characteristics in the cells, but the most important characteristics that my lab identified is in the fourth slides, atypical nucleo and atypical karyotype present in the cell. It's very important to us who works with cytopathology to identify this difference between this nucleus, because we need this information to classify the sample. Next one. Here you are. So, there is a concordance between microscope analysis and my lab. Both showed LCL. case, low-grade squamous intraepithelial case, and the focus were in choriocyte cells and the morphology of the nuclear membrane. Next one. I think this is the more important case as you need to discuss. The microscope analysis show to us a normal or a reactive case, but my lab showed the importance to have better imaging from the nucleus and classify this nucleus in this slide as a typical nuclear in the mature cells. Don't classify with LCU, but came to need to pay attention to identify this different size and color of this nucleus. Next one, please. My lab suggests the classification of this case as ASCUS, atypical squamous cells undetermined significance, not characterizing intraepithelial lesion. It's important to the segment of the patient. We need to understand in what phase the patient has the lesion and what kind of directing and treatment she needs to go on. The characteristics of the my lab present to us were mature squamous cells with a typical nucleus, showing moderate increase in the nucleus to the cytoplasm reaction. Mild hyperchromatin, well-distributed chromatin, sometimes present in small clumps or chromatin deposit in the nuclear membrane. Regular membrane, sometimes irregular contours. Regular perinuclear halo, not characterizing poilocytes. In my opinion, this is the most important case to show the borderline in negative or positive forms. Next one, please. What kind of conclusion we can show to you in this afternoon? First one, they use the EA in morphological diagnostics as proven to be an excellent tool to aiding in the identification of cellular alterations, increase the accuracy and the sensibility of the method to identify atypical cells. The equipment proves to be efficient in the preparing and analysis of cellular samples, previously prepared by automatic equipment. The important thing, you don't have a large space to install your routine. You need a single space to do it with more comfortable work. The morphological analysis performed by AI was more effective in identifying seals compared to the microscope analysis, prevalent 2 cases of LCUs and 1 case of ASCUS, previously classified as a negative for lesions or malignancy. The equipment shows promising ability to identify endocervical cells and repair the seed. Identify repair alterations is very important to the cytopathologist to do the difference between lesion or negative case. The sample characteristics of a negative exhibit evident reactive alteration of inflammatory process, being identified as negative for intraepithelial lesion, ensuring safety in cytomorphologic diagnosis, and avoiding the occurrence of false negative. In my opinion, this is the most important thing when we can identify certainly the type of the cellular change, because you need to difference the negative and the positive cells, and you exclude a high number of false negative diagnoses. Next one, please. Well, in our analysis, we understood that one of the best way of to use the MyLab is to use in a point of care model, allowing the equipment to be used in clinics, hospitals, next one, please, or healthcare centers, and monitoring post-medical produce treatments such as surgeries and lesion to check for the possible recurrence. Another important thing is use the HPV test together. Now, the proposal of WHO, World Health Organization, HPV test is a new screening guideline, and you need to use cytopathology to confirm possible lesions in the case of high HPV presence, like HPV 16 and 18, 31, and 45. I think you can use the both methodologies together, promoting great comfort for the woman when you collecting samples and obtain accurate and complete results. Next one. Here in Brazil, there is a lot of population who lives so far from the big centers. And there are a number, and this population don't have access to health service and primary care, requiring a medical service to use where these people and the luggage these people live. The indigenous, the riversides, and the quilombolas populations, even the incarcerated women could benefit from this technology with procedure performed by healthcare professionals. Next one. We suggest a workflow to use MyLab in the practical clinic inside the medical office or inside the ambulatory or the medical center far from the big cities or the big centers. First, you can prepare, suppose the patient should the medical appoint at 3 p.m. She comes or she arrives to the clinic for the appointment 1:30. She will be preparing to do the collection of cervical samples by a doctor or assistant. After this, 50 minutes after this, we will proceed the HPV test and start the preparing for the basic liquid-based cytology using the Coplast equipment and the MyLab method. We will prepare the MyLab, running the test, and around 1 hour 40, 1 hour 50, the images will be sent to the cytopathologist to do the review and to do the report to the doctor. And 5 minutes before start the meeting with the gynecologist, the patient have the HPV test and the pap test, the report to show to the doctor and to do anything as he needs. I think it's okay. One more time, I want to say thank you to the Monique and to the NOUL group to participate this webinar, and feel free to ask anything as you need. Thank you so much.
Thank you very much, Dr. Zonta, for your informative presentation and also the very nice cases, and I think important or interesting for our participants of the webinar, how it actually fits into the Brazilian market or how it could actually fit in with their markets as well globally, but also to the value points. And I think the value points you highlighted, especially in the case that you presented, that using MyLab, Sir, with the AI classifications really helps distinguish the borderline cases between NIL and ASCUS. So very, very nice cases. So we do have some questions that have come in already from the audience. But before I do that, I actually would like to introduce you to Nare Lee. She's our strategy and planning manager from our HQ team. So she's here to also to support with the Q&A sessions. So thank you very much for joining us too, Nare. So we have a question here from Dr. Glenn Dixon. So welcome to you, Glenn. Um, so he's asked for you, Dr. Zonta, how did you validate use and interpretation of MyLab stain versus your standard PAP stain?
I think my lifestyle is similar than the pipe stain. The difference is between you can standards the methods. You can use MyLab. The method is very clean, and the smears show a good images to us. Different between the traditional method or the conventional method of Pakistan, that you have a lot of interference in the smears.
Okay, thank you very much. So in terms of comparing the PAP stain to MyLab cervical, do you see any issues did you see or notice any issues when reviewing the MyLab stained slides compared to conventional slides? Was the difference problematic or did you notice that much difference?
It's not, no, no, it's not quite different. But when you analyze the image in a MyLab smear, the image is clear. It's very easy to identify the changes. And more important, the methodology can isolate the cells as you need to evaluate. Different to use the microscope, then you can screen all slides and you spend more time and maybe you can make some mistakes in this analysis.
Okay, thank you very much. And Dr. Dixon, I hope we've answered your question sufficiently. If there's any follow-ups, please feel free to put it in the chat. Um, so we have another question here. Um, I apologize if I don't pronounce your name correctly. It sounds like you're from Greece, so welcome from Greece. Eleftherios has a question. It works only with one sample per 45 minutes. So maybe Dr. Zonta, I can answer that one if you wish, or would you like to answer?
Please,
Yeah.
please, please,
So
Monique.
thank you. Thank you very much for the question. So yeah, at the moment in terms of throughput, um, so don't forget that everything's happening within this time frame. So we're doing the automatic PAPS, gold standard PAP staining, the digital imaging, the AI analysis, and the reporting out, where you can select the images and you have a PDF output, which is actually low storage capacity. So we're doing all of that with an average of 25 to 30 minutes. We are in development at the moment. So we know there is questions from the market already from our voice of customer that we're already in contact with. So we're hoping to reduce that in the future. So not 45 minutes, but actually 25 to 30 minutes on average. So thank you very much for your question there. Um, so we have another question as well from the same customer. Uh, so it detects inflammatory signs, organisms, Candida, Gardnerella, or other organisms. So maybe Dr. Zonto, because it's a development, would you like to add something before I answer? Because I don't want to take all the answers from you.
Yeah, yes, you can see the agents in the background of the smear, but my lab can't identify. I think is the next step to introducing this methodology. But you can see these agents, Candida, vaginosis, when you evaluate the imaging in the MyLab.
No, thank you very much, Dr. Zonter, and much more value actually coming from you with the experience, of course, in the field. And then from a NOUL perspective, yes, as Dr. Zonta nicely introduced, that as you know, as part of the Bethesda classifications, infectious organisms is part of that. And it is in our development pipeline to bring in also infectious organisms flag, including Candida and Gardnerella, but this is in the pipeline. So that's will be coming with next versions of software in the future.
Yes. One more observation, Monique. Sorry about that.
Of course.
We need to intend the aims of the MyLab is identify change alterations. The principal main aims of cytologic analysis is to screen if there is or there is not a change in the cells. The agents is a bonus that SMER showed to us, but the principal aims is to identify the morphologic changes in the SMER. Is the principle of Papa Nicolau techniques.
Yeah, thank you very much, Dr. Zonta. Okay, so the questions are coming in very thick and fast. It's fantastic. Um, okay, so we have another follow-up question in terms from, oops, my screen just moved. From Mostafa Darwish. Sorry, I apologize that I misunderstood your question. So the question was actually about the time from taking a sample to result. So I think maybe we can go back one slide, please, Sun,
Mm-hmm.
in terms
Yes.
of the workflow and the example that Dr. Zonta shared. So I think,
Yes.
Mustafa, this is the question that you had. So maybe Dr. Zonta, you can explain again in terms of from sample collection to result.
Okay. The time is around 1 hour, 50 minutes, 1 hour and a half. It depends the medical procedure. It depends if the my lab is near the medical office. But to do or to execute the method, you spend around 130, 140 minutes.
Yeah, thank you so much. And I
The
think this,
result, oh, sorry, Monique, sorry. The results you can send in the same time when you analyzing the image at the MyLab, in the MyLab. The same time you can produce the report and view the images that MyLab show to us.
yeah, thank you, Dr. Zonta. And I think you raised a very good point, which maybe we didn't touch on so much, but with the MyLab system, we also have an app where you can open that up on a screen, and the cytopathologist, if on the same network, does not need to be at in the laboratory or the site of testing. They can be somewhere else as long as they're on the same network for remote diagnostics within the same network.
Yeah.
Okay, so okay, so we have a question here again, comes with the equipment. In terms of subscription fee for software for AI. So, in terms of these discussions for your region, we will follow that up with you one-on-one after the webinar. And of course, if you have any questions with regards to costs and pricing for your region, please reach out to us independently, and we'll be happy to answer those questions for you independently of where you're based. So we have another question from Nigeria here, from Dr. Maureen. Oh, apologies if I get this incorrect as well. Umie Kulawuhu from Nigeria, LMIC, so low middle income country. A warm welcome to you as well. Again, a question about the cost. So, what is the cost implication for this product? So, again, we will reach out to you directly. Our business team and also sales team will reach out to you to give you the pricing for your region. Um, okay, we had actually original question in terms of validation from Sue. Me who, welcome to you as well. Is it difficult to accept just 10 samples being sufficient for a validation? So, in terms of that, it's a very good question. Um, this project with Dr. Zonta was actually an additional project away from our normal validation. So, Dr. Zonta was validating for an additional project with us, which is what he's presenting today. In terms of the validation, we obviously did our internal validation and we launched into market the product at the end of last year. And currently ongoing, we have further valid in-market validations ongoing in Latin America and also in Europe, and some pilot projects in the pipeline for us to gather further validation data with a larger sample number. So I don't know, Dr. Zonta, if you want to comment a little bit more on that.
Yes. We are starting a new project here in Brazil using my lab and HPV test as a pocket option. We will start maybe next March. You need to discuss with NOUL and show the proposed. But you are right, 10 cases not sufficiently to prove that the technology could be introduced now in the normal routine. But I think this is a promise, is correct? This is a product that soon as possible will be in the clinical to do more agility in diagnosis to the clinician and to offer more comfort to the woman when she visit the gynecologist.
Well, thank you so much. We have some really interesting questions coming in. They're really good. Um, so, in terms of another question here, Kerry Lee Francois, I hope I also pronounced your name correctly. Um, yes, you're welcome as well. And yes, she asked, can the sample be run by healthcare professionals without prior laboratory training? It's a very good question. So I'll hand that over to you first, Dr. Zonta.
Yes, we can. We can use this method in a private laboratory to see. Yes,
Yeah, and
is
I
that
think, go ahead. Go
sorry?
ahead, Doc. Go
Go
ahead.
ahead, go ahead, Monique. Yeah.
Yeah, just to add on your point, I think it's important to know, and I think you also, maybe I can ask you another question, Dr. Zonta. In terms of the training, how easy was it for you and your staff to
Okay.
run my lab and to be able to use it in terms of ease or difficulty, for example?
There is no difficulty to training my team. We need to have a team that has experience in cytopathology. And when if you have this team, the treatment is very easy and very quick.
And do
It's
you
not
think,
much. It's not different to treatment when you introduce the liquid basis cytology in the laboratory. It's similar treatment.
and do you think that healthcare professionals without prior laboratory training would find it easy to use my lab?
Yeah, it's very easy to use my lab. Yeah, you can find a lot of professionals. Here in Brazil, it's very common professionals working in cytopathology, not only as cytopathologists, but you have a lot of other professionals working in this field. And it's easy to find people to work with cytopathology techniques.
Thank you. Yeah, and from our team at NOUL, they're extraordinary in terms of development with the product, and it has a very easy user interface and user experience process. So another one for you, Dr. Zonta. Um, AI gives diagnosis for endocervical cells by Bethesda. For example, AGC favor neoplasia or AGC, NOS, etc.
É, é, the my lab identified the nuclear and the cytoplasm change. It's not defined the type of the lesion. You need to use the cytopathologic expertise to identify and to classify the lesion. But it's very important to use the EA to identify the morphologic changes. And my lab can identify the glandular cells alterations.
Thank you so much. So we have another question here from Maureen. Apologies if I got your surname incorrect last time. So she's a doctor working in Nigeria. So she's asked, has this been tested in LMICs, particularly in West Africa? We would also like to explore potential collaboration, as she's the president of Medical Women Association of Nigeria. So thank you so much for your interest. We will definitely follow up with you after the webinar, but at the moment we don't have in market experience yet in LMIC, so we will reach out to you and discover the possibilities of working with you in West Africa. Okay, so in terms of we are interested to launch, so Luis Ramon Rodriguez mentioned they're interested to launch such a test here in the Philippines. How do we start? Well, Luis, I love your enthusiasm. Again, we will reach out to you after the webinar and we will follow up with you about the opportunity there in the Philippines. So we have a question here from Barbara Herzog-Lip asking about QC. How does this work? Do you have an internal QC and an external QC for the system? So potentially, Dr. Zonta, if you could give your input in terms of Brazil, and then I can support with the rest of the globe what we've researched so far.
Okay. QC, what it mean QC?
Yeah, quality control. I apologize. So
Quality control. Okay. Yes, that is an important program of quality control in cytopathology here in Brazil. Two societies of health introduced this type of program. And I think the AI and my lab is important tool for using in quality control process because you have more exactly vision about the change, the cellular change. Maybe my lab and the EA could be a tool for use in control quality programs. Okay.
yeah, thank you so much, Dr.
Now,
Zonta.
now here in Brazil, we don't use EA in the quality control programs. We use samples, normally samples that the laboratory receives these samples, analyze, and send the report to the central.
Thank you very much, Dr. Zonta. And just from my side, in terms of answering from a NOUL perspective, at the moment, we're not producing a quality control with the device, but we know that from our voice of customers, such as yourself, that this could be needed in the future. So we have actually explored possibilities about QC in terms of already existing external QC programs. So this is something that we know might be needed, but I would be interested to continue the conversation with you for your market in terms of external QC. So I think Dr. Zonta touched on it as well. So there is a QC that's needed at the moment in some external quality control programs in terms of QCing the staining. So of course, my lab does the staining and the AI analysis. And from the discussions we've had so far with external quality control bodies, that we know that they are now looking into starting external quality control programs, including AI diagnosis as well. So I think this is also coming. So for us, for my lab, it's to the staining and the AI. So that's what we know so far. So if anybody has any information to share in terms of your needs, please feel free to also share that with us, which will also help us build out the external QC programs for AI. So we have 5 minutes left, so I'll try and get as many questions done as possible. Um, so very nice comment from Godwin. I wish this seminar will continue quarterly. So I have some good news for you. We will run a webinar series this year. I think the plan, correct me if I'm wrong, Gwen, will be that we will run 3 sessions for cervical. So not quarterly, but we will have some more coming for you. So please keep in contact and we will share that with you when it's available. So we have some more questions here. A lot, actually. So we have a question actually from Sue very early on. Another one. How many images are presented by my lab, sir, from each case to the cytologist? So Dr. Zonta, would you like to comment on that first?
Yes, it depends the number of change we have in the field, but normally more than 50 or 60 fields are showing to us to analyze the cytology.
Yeah, thank you very much. And I think you very nicely explained it's really dependent on each sample, how the LBC prep, the number of samples, the number of cells in the LBC prep, the quality of the sample, for example. But on average, as Dr. Zonta said, around for high risk and low risk, we're looking at around 10 to 50 images. And for normal cells, which is negative or flagged cell images for review needed, it could be up to 100. So yeah,
Uh-huh. It's depend of the type of the change in the cells.
yeah, thank you so much. So I hope that answered your question, Sue. In terms of the next question, there was one about how did you validate use and interpretation of MyLab stain versus your standard PAP stain? Oh, that was from Dr. Dixon. Yes, thank you. We already answered that one. Um, okay, so there's quite a few questions here, so please be patient with me to read through them. Okay. AI, yes, question take. I think we're almost answered all of the questions. Um, so I think maybe there's another one here in terms of the value, and I think you presented this very nicely in your presentation, Dr. Zonta, but in terms of the value that the value points or what was the most value for you that my lab offered you in your institution or for the women, the female population in Brazil in terms of cervical cancer diagnosis?
Well, in my experience and opinion, the security to analyze the cells is very important to a cytopathologist. My lab has a good resolution when it select the images and offer to the cytopathologist to analyze. Second point is the practicity to do the workflow to prepare the smears, to prepare the stain, and to prepare the slides. And first of all, you can send to a colleague to discuss about the case in real time, and you don't need to stay in the lab to analyze these images. You could analyze in a cell phone or in other place that you are working, not obligatory inside of the laboratory. I think my lab is not the future, is the present, because the AI is the most important discussion in our work. and on what type of use you can introduce AI to help physicians, to help pathologists, or to help health in complete. My lab is an important tool that we use for population who lives far from the big cities or far from the medical centers, the important medical centers. And well, if I I send a note, uh, 0 to 10 to my lab methodology, the point is 9. It's a good. We need a lot of appreciation or modifications in some points, but it's a resource. It's a very good resource to use to screening and to the diagnosis cytopathology. I think we need to test to use not only in cervical cells, but in other types of cells, like thyroid, like breast, like liver. I think this equipment or this methodology will be very important when you use the fido needle function uh, for the analyze the cells in hospital or in urgent service.
Oh, thank you so much. And I think we've come to the end of the webinar now. So, for those questions that we didn't get a chance to answer, I've seen a few more come in now that we will get back to you after the webinar. And a heartfelt thanks and gratitude to you, Dr. Zonta, for your participation in this webinar today. You can reach out to this email address here, marketing@noul.com, if you would like to ask your question. Apologies that we didn't see your hand earlier. But again, thank you everybody for joining us. Your participation is incredibly valuable for us. Your questions today, very fruitful discussions. And whichever questions we didn't get to, we will follow up with you one-on-one. And of course, if any questions arise later on, feel free to reach out to us. I would also like to thank, of course, Dr. Zonta. Thank you so much for your insights and your experience and the cases, real world examples and insights and experience of MyLab Cervical and how that's really impacting you and your team, your workflow, your ability to have confidence in the diagnosis and be able to see ascus over nilm from manual microscopy, and how you're really contributing to the community and reaching this goals of the WHO by 2030. Let's say we're all contributing, and nicely coinciding with Cervical Cancer Awareness Month this month in January. And before we leave, I would also like to say a special thanks to the team at NOUL for all of their efforts at organising this incredible webinar. Gwen Park, Nare Lee, and Sun Choi. Without you, this would not have been possible as well, with all of you, Dr. Zonta and the team at HQ, and also all of you in the industry, our distributors, our potential customers all over the globe. Thank you for your interest and your participation today. And as soon as the webinar's closed, a window will pop up for a survey, and it'll only take 1 or 2 minutes of your time, and your feedback is incredibly valuable for us to, let's say, make changes for the future webinars, or to understand the value points, or potentially also what we could focus on in our next webinar. So thank you so much. And if you participate also too, you will also receive the recording of the webinar as well for looking back and sharing with your colleagues. So thank you so much. And I know it's middle of January, but from all of us at NOUL, we wish you a successful, happy, healthy, and fun new year. And yeah, have a beautiful rest of your day or good sleep, and see you next time. Thank you from all of us at NOUL, and thanks again, Dr. Zonta. Thank you.